Okay guys, this video, I just want to take a few minutes to kind of go over a couple of things. So you have a test coming up. And so I want to talk about what's going to be on the test, and I also want to talk about the lab quiz that you're going to take over diffusion and osmosis. So this isn't too fancy or anything, but I just wanted to go over just some highlights. So first off, the test, there's going to be a total of 3 sets of notes on the test. You did have a study guide. There's not a whole lot in it, but there are some practice questions. So the main thing you need to study are the 3 sets of notes. And so the first one are your cell notes, and that's where we talked about things like cell theory, the size of cells, the types of microscopes that we use. We compared prokaryotes and eukaryotes, how they're alike and how they're different. We looked at the endosymbiotic theory, which kind of explains how the eukaryotes evolved. And then we looked at just some general internal membranes and why those might be important and make the cell more efficient. Now the second set of notes was actually not a PowerPoint, it was actually the activity that you did. So you had a packet about cell parts and organelles. I will tell you that there are no diagrams on the test. So you won't have to identify them by pictures, but you will have questions about the cell parts and their functions and their structures. So essentially that big chart that you filled out with the matching activity. And there could be some questions about, the endomembrane system or anything that was on there. So just make sure you look over that set of notes or that activity as well. And then the third set of notes was the cell membrane and transport. And so we had a quiz over the cell notes and the cell parts. We did not have a quiz over the cell membrane and transport because instead you had a lab. And so in the cell membrane and transport notes, we talked about membrane structure, we talked about how the membrane is selectively permeable, and we talked about different types of transport, primarily passive transport, and those include diffusion, osmosis, and facilitated diffusion, so you need to know what each of those are. All of them are passive, which means they're high to low, and then you have active transport, which is going to be low to high and requires energy, and that includes your pumps, that includes co-transport, and that includes the bulk movement like endo and exocytosis. So this is just kind of a general breakdown. I could have left something out, but for the most part this is just going to be everything that's on those notes is game for test. And then you do have an essay on this test. It is in your study guide and it is a chart, and I actually explained it when I explained osmosis. And so essentially the whole setup is there's a blood cell. It's going to be placed in 3 different solutions, and it gives you the concentration of those solutions. And so you got to fill out the chart, and it's going to be like the last test where you're going to take the multiple choice portion, and then once you're done with that, you will be given the chart. Okay. So you're going to, in one column, you're going to identify the percentage of the solution, and you're going to determine whether that's hyper, iso, or hypotonic. In the middle column, you're going to tell what happens to the cell. That's essentially going to be like the cell is going to expand or swell or possibly burst. The cell is going to stay about the same, and the cell is going to shrivel or shrink. And then in the third column, you need to explain why based on osmosis. So in that third column, you're going to be talking about water moving and why it's moving a particular direction. So because it's osmosis, don't just use words like it will, what will, okay? It's about water. So make sure you explain what's happening to that cell based on the water movement. And then I also wanted to go over just a little bit about lab quiz. And so your lab quiz is actually going to cover the 3 parts of your lab. You had questions on your pre-lab. You also had questions in the lab packet. And these are the same types of questions that you will see on the lab quiz. So the lab quiz is just to make sure that you understood big ideas of the lab. And so just some general things you might want to focus on. So part A was diffusion across a selectively permeable membrane. And so that's the one where we kind of did as a class demonstration. And so what you're going to do is you need to understand kind of, first of all, what is selective permeability? Was the membrane selectively permeable? The answer is yes, it was. And the reason why is because some molecules did move through the bag and some molecules did not move through the bag. So you need to understand which molecules moved and which ones did not. Now, did we test for all molecules? Technically, the answer to that is no. There was one substance that we did not test the movement of in part A, okay? So you just kind of be familiar with the idea of which molecules moved and which ones didn't, but we did not technically test for all molecules. And so some were able to easily pass through the bag, and we were able to identify that through color changes, and some molecules were not able to pass through that dialysis tubing. In part B, we had different sucrose solutions, and we narrowed it down to 4, but essentially you had sucrose solutions, and sucrose is a disaccharide, so it's a little bit bigger of a sugar than glucose. But sucrose solutions varied from 0 all the way up to 1 molar. And so we had a 0.2 molar, you had a 0.6 molar, and you had a 1.0 molar. And then you had just plain old distilled water, which had 0% or 0 molarity of sucrose. And so what you're looking at here is just the movement of water by osmosis. And so you were trying to answer the question, how did the concentration of sucrose affect the movement of water? So I hope that went smoothly yesterday, and I hope you got to see some good results. So you need to understand which bag changed the most and kind of why, which bag changed the least and kind of why. You did measure in percent change in mass, so those are the values that you're kind of basing that off of. Since they weren't all the same to begin with, we need to do a percent change. And so just kind of understand the movement of water based on different concentrations of solute. And then part C was water potential. Now in the actual lab packet, it describes the procedure. It's very similar to the video that we watched. The only difference was in the video, we looked at length of the potatoes. In the actual lab, we would have measured the mass and done percent change like we did in part C. But basic idea was we took potatoes of like potato cores, and we placed them in different concentrations of solutions. In the video, they used salt. In the actual lab, it was sucrose, but just understand that it's just a solute concentration. And so you do need to understand that the potato does have its own concentration of solute. And so you need to understand, why did some potatoes increase and why did some potatoes decrease? And that's based on water potential, okay? And so you should also know how to determine the concentration of that potato. So they showed you on the video about the graph, how you can determine the concentration of the potato. And then water potential in general, just know what it is. Water potential takes into account more than just your solute concentration. It also brings in the concept of pressure and some constants. Essentially, we use it for water movement in plants. And you do need to be familiar with, in terms of water potential, how does water move? And so kind of like osmosis, it's going to be from high to low. And so high water potential means it's going to have more water and less solute, and lower water potential means it's going to have less water and more solute. So just make sure you kind of read through those. You can always watch the videos again, but for test and quiz, these are the things that you need to be focused on. Definitely prepare for your essay, and if you have any questions, just let Ms. Petty know, or you can email me. Just make sure you prepare. Okay, this is the second test for the 9 weeks, and it is going to be your last test for the 9 weeks. So you want to definitely make sure that you are prepared and you do well. Hope you guys have a great day, and I'll see you soon.